Hello and welcome everybody to my channel Omni Bioentrance, and today we will be discussing CSIR Net JRF Life Science question paper. These are questions of the shift 1 or the morning shift question paper, and this is part C I will be discussing. And in this part C I am discussing applied biology as well as methods in biology, and this is the second part that I am doing. And this exam was conducted in March 2025. So before coming to the next question, I would like to know how these proteins get localized into the endoplasmic reticulum lumen. It is nothing but your SRP and SRP receptors. So just to brush up, I am telling you this translation, it begins in the cytoplasm. So the proteins which are destined to enter into the endoplasmic reticulum lumen, they will have a signal peptide sequence at the N terminus, remember that, and this sequence it will be recognized by the SRP. The SRP binding will pause the translation, and the SRP will get bound to the ribosome, and it will dock the ribosome and the protein, nascent protein, to the SRP receptor found on the endoplasmic reticulum membrane, and there is GTP hydrolysis and all those steps taking place, which will result in the dissociation of SRP, and the ribosome, it will get associated with the SEC61 translocon complex in the endoplasmic reticulum lumen. And this translocon complex, it forms a channel, and the newly synthesized protein chain will directly enter into the endoplasmic reticulum lumen. Now the translation will continue, and as the translation continues and the protein chain grows, the signal peptide sequence at the N terminus will be cleaved off by the signal peptidase. And once it is cleaved off, the protein enters into the endoplasmic reticulum and undergoes folding or post-translational modifications, etc. So this is what is happening, but remember the point, at the N terminus you have got the signal peptide, and it will be cleaved off once the protein enters into the endoplasmic reticulum lumen by the signal peptidase. Remember that point. Okay, now next question. A researcher needs to identify if protein A is localized in the endoplasmic reticulum in a human cell line. Okay, so the researcher has identified a new protein called protein A, and he or she wants to see whether it is localizing in the endoplasmic reticulum in human cell line. So for that, what he or she has done is, he has tagged the cell has an RFP tagged protein that marks the endoplasmic reticulum in this cell. So first he has to use some fluorescent marker to identify endoplasmic reticulum. Okay, so he has used an RFP tagged protein found on the endoplasmic reticulum. So when it gives fluorescence, it is endoplasmic reticulum. Okay. Now what the following are the proposed experiments. So these are the various experiments that he has to decide which one to use to see if protein A is localized in the endoplasmic reticulum. So first what he has done is, he expressed protein A fused to GFP at the N terminus in the cell, followed by microscopy to check for co-localization with RFP. So what is his intention is, he will tag the N terminus of protein A with GFP, and then he will use fluorescence microscopy, and he will see whether the protein A is whether he get a signal of protein A as well as the signal of RFP tagged protein in the endoplasmic reticulum lumen. So both these signals, if he gets, he can confirm that the protein is localized in the endoplasmic reticulum lumen. So what is the use if he tags the GFP at the end terminus in a cell? He is not going to get a signal. Why? Because once the protein enters the endoplasmic reticulum, end terminus, I told you, it is having a signal sequence. This signal sequence will be cleaved off by the signal peptidase. So if it is at the end terminus, signal peptide is there, signal peptidase will cleave, GFP also will be cleaved. He will not get any signal. He may get only the RFP signal, but not GFP signal. Okay, so A is not correct. So that makes 2 wrong as well as 4 also wrong. Now next one. Express protein A fused to GFP at the C terminus. Yes, then he will get both the signals. That is, he will get the GFP signal from protein A as well as RFP signal from endoplasmic reticulum. So he can conclude that protein A is localized in the endoplasmic reticulum. So B is correct. Then next is immunofluorescence staining of A followed by microscopy to check for co-localization with the RFP. Now what is this immunofluorescence? It is a microscopic technique and is used to visualize as well as to locate proteins within cells. Now in immunofluorescence, what you generally do is you have got an antibody conjugated with a fluorescent dye, and it is allowed to bind to your protein of interest, and then you view it under the fluorescent microscope and it will emit fluorescence once the antibody gets bound to your protein of interest. So here what you will do is if protein A is localized in the endoplasmic reticulum, by using this fluorescently labeled antibody, you can find out how this antibody, it will bind to A and it will emit fluorescence. So here endoplasmic reticulum also you will get one fluorescence, and this protein A also will send a fluorescent signals. So when you get both the fluorescent signals, you can confirm that A is localized in the endoplasmic reticulum. So C is correct. Oh sorry, I made a mistake here. A is not correct. So 1 is wrong and 3 is wrong. Okay, B, C. Okay, we will see whether D is correct. Okay, B is the right statement. I made a mistake there. Okay. Isolating the endoplasmic reticulum by differential centrifugation and checking for co-purification of protein A with RFP. Okay, now what you do, you will isolate endoplasmic reticulum. You isolate the protein A, you isolate protein which is bound with RFP, and then you check for the co-purification of protein A with this RFP tagged protein. You find that they are co-purifying, that means protein A is localized in the endoplasmic reticulum. So D is also correct. So B, C, D are correct. That makes option 2 as the right answer. 2 is the right. The fourth one they have given only B and C. D they have not mentioned. So fourth is wrong. Then next one, transcranial magnetic stimulation. What is that? It is a non-invasive procedure. So here what you do is you place a magnetic coil. Here you can see this is the magnetic coil. This is the instrument. It is attached to a computer. So this is the magnetic coil that you place on the scalp of the patient. Okay, and it generates high intensity magnetic pulses. And these magnetic pulses, here you can see they generate a magnetic field over here in this region of the brain. And these magnetic pulses, they create induced current in the brain or electrical current in the brain in the specific targeted area beneath the coil. So the coil is placed, this is the targeted area, in such a way it is just beneath, it is just above the targeted area you place the coil. Okay, this is the targeted area in the brain. Above that you place the coil. Then it is attached to a computer. So through the computer you can adjust the frequency, intensity, duration of the magnetic pulses. Fine. And this helps the doctors to stimulate or you can even inhibit specific brain regions and study their various behavioral characteristics. What will happen if you stimulate this particular brain region? What will happen if you inhibit this particular region? All these things you can study. And TMS, it is usually used to treat depression and OCD. OCD is obsessive compulsive disorder. So all these things TMS is used. Now come to the question. So the magnetic field generated from an electromagnet is used in the transcranial magnetic stimulation of the brain. The following statement suggests some features of the TMS. Which are the correct one? The magnetic field generated in TMS induces an electric field in the underlying brain area. I already told that. So that is correct. So A is correct. Okay, that means second gone, third gone. The electrical field in the brain area alters the membrane potential of the neurons in that locality, causing them to depolarize synchronously, which in turn may change the probability of the firing neurons. Okay. So in this TMS, you have the magnetic field and it induces the electric field in the brain. And because of this electric field, the neurons in that targeted area, they will change their membrane potentials in accordance with the electric field, and this will result in their depolarization synchronously in that particular area. So that is correct. So that half is correct. That is, electric field in the brain area alters the membrane potential of the neurons in that locality, causing them to depolarize synchronously, which in turn may change the probability of the firing neurons. Now what is this probability of firing neurons? That means it can either increase the probability of action potential and make the neuron to more likely fire. Okay, or it can decrease the probability of action potential and make the neuron less likely to fire. That is what it means by change the probability of the firing neuron. So this TMS and this electrical field in the brain area, it can depolarize synchronously. It can either increase the probability of the action potential or decrease the action potential. That is what is meant by statement B. So B is also correct. That makes option 4 also wrong. Then third, in cognitive neuroscience research, TMS can be used as a tool to induce a virtual lesion in a selected region of the cerebral cortex. Okay. Now TMS, I told you, you place a coil on the scalp and it generates a magnetic field, and the magnetic field passes through the scalp and it induces an electric current in the underlying brain area, and this induced electric current, it will depolarize the neurons in the targeted area, and this depolarization is temporarily and because is temporary, and by observing the effects of this temporary depolarization or temporary disruption, you can study these behavioral changes in that region of the brain. That means if suppose a person is epileptic and you find that it is in the cerebral cortex region, so first you place the TMS coil in that particular region, and you pass the magnetic field, then electric current is produced, it depolarizes the neurons and it will temporarily disrupt some neurons in that particular region. Okay, that is, it creates a virtual lesion. So if it was the original type of lesion, means you will have to operate the brain, then you have to do the damage to that particular area and then treat. It is going to be very difficult. Nobody will appreciate that kind of treatment. So if using TMS means you can create a virtual lesion, you can temporarily disrupt the function of the neurons in that particular region, and you can see whether the disruption has resulted in a decrease in the epilepsy of the patient. If it has decreased, okay, fine, this region of the cerebral cortex is responsible for the epileptic behavior of the patient. If it is not, you can just move the coil to another specific region in the cerebral cortex, disrupt the neurons over there, temporarily disruption, and then you learn whether that region is causing the epileptic behavior in patient. Like that, you can study by creating what is called as virtual lesion in the selected region of the cerebral cortex. So that was just an example for you to understand. Okay. So C is correct. Then TMS is safe and non-invasive. That's 100% correct. But the neuronal activity of the stimulated area is disrupted for a long period. No, I told you it is temporary disruption, not for a long period of time. So that is D is wrong. So A, B, C is correct. That makes 1 as the right answer. Okay, then coming to the last question. A purified 150 kDa protein species from gel filtration column. Okay, gel filtration means the purified protein will be in the native form. It will have its subunits perfect. That is, if it is a dimer, both the units will be perfect. If it is a heterodimer, then both the subunits will be again perfect. Multidimer, all the subunits will be perfect. So without any disruption of the subunits, you will get the protein in the native form. Was run on a two-dimensional SDS page. Okay, so here the two-dimensional SDS page is such that one dimension you are running SDS page and in the other direction you are running SDS page plus DTT. Now once you see two-dimensional SDS page, don't think it is your first dimension is isoelectric focusing and the second dimension is your SDS page. No, here in the question it is first dimension is SDS page and second dimension is SDS page plus DTT. So whatever you know regarding your what is that? Isoelectric focusing and SDS page two-dimensional gel electrophoresis, keep it aside for the sake of this question. Now what they have done, what is the likely form of the 150 kDa protein species from this observation? Okay, you have to see whether it is two proteins with a non-covalent interaction, two proteins with the covalent bonds, etc. You have to see. So one thing I will tell you, it is DTT. DTT, it will break the disulfide bonds between two protein subunits or two different proteins. And the disulfide bond is a covalent bond. Fine. Now what does this SDS page separate the proteins? So SDS page, it is a detergent and it will unfold the protein and it will give a uniform negative charge to the protein. And the protein will migrate in the gel with uniform negative charge towards the opposite pole. Okay, and it is this SDS page, the proteins are separated based on their molecular weight or size. Fine. That is what SDS page is doing. That means SDS page will break only weak bonds, not covalent bonds. Fine. Covalent bonds are not broken by SDS. But DTT, when you add, it is breaking the covalent bond, that is your disulfide linkage. Now come to the question. So here is the gel photograph that they have given, and for easy understanding, this side I have marked as 1, this side I have marked as 2, this side as 3 and this side as 4. This is the one that I have drawn. So this is the first dimension that you do only SDS page. Okay, so you apply your sample here. The sample has migrated as a band over here. You get a single band with SDS alone. Okay. Now you rotate the gel so that 2 comes here. Here is 2. 1 is over here. So this gel band you will find it over here. Okay, this is the gel band that you got by running using SDS alone. Now the second dimension, you treat this gel with DTT also. So if there are any disulfide linkages, they will be broken. And you will get 2 bands like this from this band. Okay. So this band will not be seen in the gel again. It has got split into 2 bands upon treatment with DTT. It has, that means there was disulfide bond between these 2 proteins, and they, when on addition of DTT, they got broken and you got 2 different bands. Fine. Now this is how the gel what they have represented after the first and second dimension. This is the gel. So these are the 2 bands that you got. Now read the observation. There are at least 2 proteins that are linked through non-covalent interaction. It cannot be. Okay, there are 2 proteins, that is why you got 2 bands in the end. But that is disulfide linkage is not non-covalent, it is covalent interaction. So that makes first option incorrect. There are at least 2 proteins in the complex that are linked through covalent bonds. That is correct. There are 2 proteins in the mixture without forming a complex. Not correct. And there is only 1 protein that has a disulfide bond. No, there are 2 proteins. So that is also not correct. So that's it. Thank you for watching my channel. If you are new to my channel, please do like, share and subscribe. Thank you.